Today I'd like to talk about how do we pass on our genetic traits. This is called heredity. Heredity. How do we pass on, how do we get certain traits and characteristics? We studied mitosis, cell division. We'll find meiosis to be very similar to mitosis, however, have a completely different outcome. We'll also study how does probability for mathematics come into this. We'll talk about how Gregor Mendel came up to this important discovery. Let's start with him. Let's start with Gregor Mendel. He worked in he was in a monastery when he was working with some pea plants. And what he found is that when he cross-pollinated the pea plants, he found that that the pea plants did something very strange. They varied. If he had two tall pea plants, so we'll make these pea plants green. He had two tall pea plants crossed. So he got one tall pea plant, one short pea plant, and cross-pollinated them. And from there, he got four tall pea plants. But then in the next generation, he called this generation the F1. So this is the parent. This is the F1. Then he noticed something in the F2 that was very interesting, which is the generation that comes after the F1. If we go down here, we'll actually just have to go down here. What he noticed is something very, very strange. He noticed he had 3 tall pea plants. He had 3 tall pea plants, but he had one that was really short. Well, now that puzzled him. He started off with the tall and a short. Okay, then he got 4 tall. Then he got 3 tall and a short. That was very, very strange. So he did some investigating and found out about heredity. We'll talk about why Gregor Mendel was so lucky to choose pea plants over other things. But let's start with heredity. If we look at certain things here. We talk about chromosomes. We have first a whole bunch of vocabulary. We have chromosomes. Remember from our mitosis. We have we have gametes. We have alleles. We have genes. We have traits. And then we have 2 long words, homozygous and heterozygous. We're just going to go through these. Chromosomes, we remember, are the structure of DNA found in the nucleus. These are structures of DNA found in the nucleus. If we draw a picture of a chromosome here, this is a chromosome. This is a chromosome. Winding up like that. Actually, yes. Then, we have a duplicated chromosome here. And so those are what chromosomes look like. Gametes. Gametes are sex cells. They are what we use to sexually reproduce. Those are the sperm and the egg. Now, some interesting things about gametes. They are haploid, meaning they have only one set of chromosomes. Humans have 46 chromosomes. 46 chromosomes. From those 46, female stones have 46 chromosomes. We get those 46 by getting 23 from our mom and 23 from our dad, to total up to 46 chromosomes. Now, that's interesting. We have 2 copies of each gene, because we have one from our dad and one from our mom. So, that's why they're considered diploid. We have 2 complete sets. There's a complete set here, and a complete set here, coming into a diploid. So, 2 haploid, becoming 1 diploid. So, these gametes are haploid. This one here has 26. This one here has 23 chromosomes. This one here has 23 chromosomes. I'm going to go ahead and skip down to genes before I go to alleles. Actually, let's start with traits, because traits seem the most easy to remember. Traits can be there are 2 types of traits. There is genotypes and there are phenotypes. Now, the phenotype, both of these are traits. These are the genotypes are the genetic traits. The phenotypes are the physical traits. A phenotype would include brown hair, black hair, blonde hair. A genotype would include, which we'll talk about later, are things such as let's think back to the pea plants. Tall, short, tall, tall, short, short. So homozygous short. And so we'll talk about these here. So those are traits. After defining that, genes are fairly simple to define. They are the factors that control the traits. So the genes control the traits. So the genes determine whether you're going to have brown hair, blonde hair, or black hair, or whether you're going to be, whether your genotype is going to be tall, tall, short, short, or tall, short, things such as that. Now, homozygous and heterozygous go back to genotypes and genes. Actually, first let's define allele before we go to homo and heterozygous. An allele is one specific variation of a single gene. So, if you look at a specific gene, let's say the genotype for tall. Tall is, if you're tall, or let's look at it for short. The short is lowercase t, lowercase t. I'll explain what that means in a little bit. But one allele of this is this lowercase t. That's an allele. The other allele is the uppercase T. So we have uppercase T and lowercase t. Now, for things such as blood types, it can get a little confusing because there are multiple alleles for blood type. There's IA, which gives you A blood, which is the antigen for A blood. IB, that's the antigen for B blood, and then just lowercase I, that's how you get O blood. No antigens, that's O blood. So you see there are 3 alleles for blood type. Now, homozygous and heterozygous go back to genotypes. You notice that this is T, T, and T, T. If you have capital T, capital T and capital T, or lowercase t and lowercase t, those are considered to be homozygous, because homozygous, they are the same type of letter, capital, capital, lowercase, lowercase, and they are the same letter. So capital, capital, lowercase, lowercase, same T. These are homozygous. Heterozygous look like this. They involve a capital and a lowercase. So capital T, lowercase t. Next, we're going to talk about a couple of new terms which involve those capital and lowercase t's. For example, we're going to talk about dominance, codominance, incomplete dominance, and incomplete dominance. So what we see here is, dominance. Capital letters are dominant to lowercase letters. If we take two capital T's, the organism is going to be tall. Remember that the T's are going to say they're genes for tall. If it has one tall and one short, we know that capital letters are dominant, meaning their phenotype will always show up. So this one here is still going to be tall, even though it has one of the short alleles. However, two lowercase t's are called recessive. So the lowercase t is recessive, meaning that it does not show up often. It's still there, but it won't show up. If you have two recessive, then it has to be short. The bottom line is, if you have any dominant in your genotype, if you have any dominant allele, that phenotype is always going to show up. That contains a lot of vocab words there. So if you have any dominant, that's the capital letter, allele, that's either this one here or this one here, this one or this one, this one or this one. If you have any dominant allele, then that phenotype is going to show up. So the phenotype being the physical trait. So if you have the dominant allele of tall, the tall phenotype is going to show up. If you just have two recessive, then that's short is going to show up. Codominance is these are special types of dominance. Let's start with codominance. Both alleles are fully expressed. So that means that if something is codominant, such as blood type, that's the number one thing. If you have AB blood, the genotype for AB blood looks like that. There's no IAB, it's just IA and IB. You might see just the A and just the B without the I's, but the convention is that you have to use the same letter. That way it's not confusing, because there are such things as polygenic traits, which basically use multiple letters, and we'll get to those in a minute. So codominance, this is IAIB. If you look at a person's blood type with codominant, like with AB blood, then you'll notice that they have both A and B blood. So they have codominant because both alleles are being fully expressed. Now, if you look at someone with incomplete dominance, you can, for example, think about, say, a pink rose. We see that we have pink roses. We know that pink roses are a combination of red roses and white roses. So incomplete dominance is where the two traits are blended. So if we have a red rose cross-pollinating with a white rose and they generate a pink rose, it's because of incomplete dominance, because both alleles are fully expressed. So, or because both alleles are blended. So R and R, that becomes pink. There's a lot of misconception about there's a lot of concern about when do we see the how do we know that something is going to be either incomplete dominance? Well, if you're given a problem like this on the test, then they should explicitly say these two are incomplete, and by that, then you know that the heterozygous genotype is going to give you a blended phenotype. Now, I mentioned before about something called polygenic. So if I look here, multiple alleles would be like blood type. AI, okay, let's actually use multiple letters. AI, AE, AO. This is called multiple alleles. Multiple alleles. Now, a polygenic example is hair color. Hair color is polygenic. Polygenic means that you have multiple letters. Oh, okay, let's actually clear it. Let's make some space here. If you have a polygenic trait, then we have A, B, C, things like that. That is polygenic because there are multiple letters. So this one's the same base letter, but different, but basically like differentiated by subscripts. And when you have polygenic, so I'm gonna write that here, polygenic. When you have polygenic, then you have multiple letters. We'll talk about how heredity comes into place with Punnett squares, and how you can predict the probability of having a certain characteristic show up.